Wonderful. Thank you everybody for being here. Really excited to present this information. As Allison said, my name is Brenden Campbell, and today we're going to be talking about multi-solving waste to food, from intergeneration impacts on styrofoam-fed mealworms to farm-scale production. I first wanted to start off with some definitions. Expanded polystyrene, abbreviated to EPS, I'm going to be referring to as styrofoam, which is the more common name for it. We also have 4 different mealworm life stages of the egg, the larval form, which is depicted here. Think similar to a caterpillar. Then we have a pupa, which is kind of like a cocoon, a transitionary phase. And then the beetle, which is depicted in this upper right-hand corner here, which is the mating stage. Frass is mealworm excrement. FTIR, ATR is Fourier transform infrared spectroscopy, attenuated total reflectance. I will refer to this just as FTIR throughout. When we go through talking about generations, I'll be talking about 3 different generations of the parental generation, or the P0, which is the first generation that was exposed to the styrofoam treatments. We have the F1, or the first phylal generation, which is the offspring of the parental, or the second generation exposed to styrofoam treatments. F2 being the second phylal generation, or the offspring of the F1, and the third generation exposed to styrofoam treatments. And lastly here, system dynamics, defined as an understanding the structure of a system to understand the behavior we observe and can see. So to dive into our problem statement and how all of this research ties in with broad scoping problems, I first want to talk about population and undernourishment. For population, the total population today as of 2022 is 7.9 billion individuals. And the United Nations has estimated that that population will rise to 9 billion by 2050, and now just under 11 billion people by 2100. This causes mounting pressure on food systems. So with recent and ongoing COVID-19 pandemic, there was an increase in the number of individuals who were considered undernourished, and previously progress against world hunger and undernourishment was improving through a 2005 to 2014 period. However, a slight uptick in those food insecurities arose in 2014 and led to a massive increase in 2020, likely due to a response to COVID-19 and subsequent containment measures that demonstrated flaws in the supply chain. 99% of undernourishment is in the Asian, African, Latin American, and Caribbean regions. Interestingly, regions where insect consumption is the largest is in Asia and Africa. Often it is insect foraging, but that is not the common choice. It is just something that is a necessity. So how can insects help? Why is it a good solution in those places? Well, it's easy to farm insects in most locations near the equators because of good humidity and temperature. And it's relatively efficient as a feed conversion ratio for converting non-human edible materials into human edible animal protein, carbs, and fats. Insects also have very good digestibility and can cover a wide range of nutrition needed for consumption, depending on what type of insect is consumed. So the most consumed insects by humans are beetles, which are depicted here, as well as bees, wasps, grasshoppers, locusts, crickets, ants, or caterpillars. Wide range of different species of insects are consumed. About 2 billion people with cultural backgrounds in the Asian, African, and Latin American areas consume insects. This covers approximately 80% of all countries, meaning that only 20% of all countries, including most Western countries, are not currently consuming insects. However, there are multiple regional and national organizations that promote insect agriculture research and bolster new business by working with governments and set regulations that will allow insect agriculture to boom. These organizations include the North American Coalition for Insect Agriculture, the Asian Food and Feed Association, the Insect Protein Association of Australia, and the International Platform of Insects for Food and Feed. Within each of these platforms and coalitions, dozens of insect industry members share knowledge and increase agricultural presence and advance government regulations. I now want to talk about the global plastic production and how it fits in with this conversation around insects. While plastics were invented in the 1860s, production did not explode until the 1940s, in which it became one of the fastest growing industries. In 1950, plastic production was only 1.7 million tons per year. And in 2020, global plastic production was estimated to be 367 million metric tons in that year. Industry analysts project a return to an average of 3.4% growth per year, and plastic production has overtaken other common materials such as glass, metal, paper. Styrofoam itself accounts for 15.6 million metric tons in 2019 and is expected to increase. Only 9% of plastics have ever been recycled, with 12% being incinerated and the remaining 79% being landfilled or lost to the environment. Plastic is everywhere. We live in a plastic world and are surrounded by them every moment of our modern lives. We need some solutions, and insects can be one of many solutions that can be applied to this problem. So a potential solution to styrofoam specifically, which is what this study covers, exclusively styrofoam consumption by mealworms. Interestingly, insect consumption of plastic material is not uncommon and can be seen in a multitude of other insect species. And not only has styrofoam been shown to be broken down by multiple insects, but so have many other types of plastic, such as polyethylene, rubber, PVC, and PLA plastics. The prospect of mealworms breaking down styrofoam is exciting, and there's potential to turn this insight into a more ecologically sustainable system. And again, this study focuses solely on styrofoam. The objectives of this study were to first determine the generational and treatment effect on nutrition composition when mealworms consume styrofoam. Let's move this over here. It was also to determine if frass can serve as a fertilizer, meaning that there'd be no styrofoam in the frass after consumption, as well as analyze and evaluate a lab-driven model for large-scale population and supply chain to determine and simulate population growth, styrofoam consumption, and profit. This is a look of how the objectives were met through each treatment. This was done by utilizing 9 different treatments, and this was consistent for each of the 3 generations. Each treatment was given a set amount of chicken feed and styrofoam and placed within tiered bins. And I'll have an image of that in a later slide here. These ratios that you can see up here were derived from another study and extrapolated upon to come up with the rates here. So for example, this is a 32 parts chicken starter to 1 part styrofoam or expanded polystyrene here. What that would look like is 192 grams of Purina chicken starter to 6 grams of styrofoam. The abundance treatment was chosen and determined as the large enough quantity for the mealworms to never run out of food. The sufficient treatment was determined as the precise amount of food the larval form would need to survive and pupate. And the blend was determined as a middle ground between the two treatments. This is a look at how each of the bins were rotated while within the system. So each day, data was collected for a total of 300 days worth of collection. And every day, each bin was counted, different metrics were counted, and they were moved one place clockwise to the left. So every single day, the bins were rotating. The humidifiers are depicted here as they were below the bins in order to create an attempt at optimal humidity and temperature. The humidity did not reach optimal humidity. It only reached between 50% and 55% relative humidity for the average of the entire 300-day period. Temperature was consistent between 77 degrees Fahrenheit and 80 degrees Fahrenheit, or around 23 to 25 degrees Celsius. This is a real life picture of the farm system and how it was set up. We have the bins on the tiered rack here, and the humidifiers are down below here. For the methods, how all of it was done, frass was collected, and then FTIR, ATR analysis was conducted on the mealworm frass, as well as counting how long mealworms were on each treatment for each generation. This metric was determined as how long did it take each bin to reach 100 pupa. Nutritional analysis on mealworms was conducted, and that was done in the form of percent crude protein, percent crude fat, and percent dry matter. Body weight, larva population, styrofoam consumption, chicken feed consumption were also all data points collected and then further statistically analyzed through analysis of variance or ANOVA testing. And this was done with the help of Dr. Makivar, or Martin, as many of us know him. Larval populations were also simulated for each treatment, as well as a simulated supply chain and styrofoam consumption for each treatment. This is a close-up picture of the bins before chicken feed was added of the mealworms on the styrofoam. I want to point out the 1/50th inch mesh that was used as a passive frass sifting mechanism. This will become important when we talk about FTI results and implications in the next two slides here. This is a close-up of an FTIR machine, if we've never seen one, and it was used to produce the following results. So initial FTIR ATR analysis determined that there was no styrofoam detected in mealworm frass for all treatments for all generations. And it's first important to detail that the collection method of frass, which involved that 1/50th inch mesh separator, and the fact that frass sat for around 3 to 9 months before being analyzed. So this FTIR spectra was compared with industry software to analyze if there was that presence of styrofoam. And that's what can be seen here in these two lines of matching the black, which is the frass treatment, and then the orange is the industry software matching it and coming up with what substances it most closely matched to. The most commonly matched substances of the frass samples were coconol, which is a type of insect scale or exoskeleton, as well as trisodium phosphate and pancreatin, which is commonly found in a lot of feed additives. This differs from other research in which only 50% breakdown of styrofoam was found. And so there is a possibility and high likelihood that plastic degradation and those microorganisms associated with that could be ingested with the frass, and that they could continue their breakdown over time. The previous collection methods of frass were in a 24-hour period shortly after consumption to ensure a timely collection and analysis. However, like I said, in this study, it was done 3 to 9 months after, depending on which generation it was. So there could be a time element added to the breakdown of styrofoam that should be further investigated. Answering if there is styrofoam enzymes or other enzymes responsible for breakdown post-egestion could help answer if they the breakdown post-egestion could be responsible for a total breakdown of styrofoam that wasn't seen in other studies. This could also help explain why there are a handful of studies that have indicated no finding of styrofoam in the frass. And this difference in collection method could be one of those explanatory factors. It's a little bit hard to see at the top here. So when talking about how long it took each generation to reach that point of 100 pupa, the first generation was received at a medium size. So they were already halfway through their life. So they only took 40 days on average to reach that 100 pupa mark, which is statistically different than the F1 and F2 generations. Those generations started from egg all the way up to pupas, where the time was calculated, and the trend of time that it takes was decreasing between the F1 and the F2 generation, enough that there was generational statistical differences. Further research would help show if this trend continued and if there is a plateau point for the feedstocks and treatments that they were given. To now touch on nutrient analysis. First, analyzing the average mealworm percent crude protein and the effect that generation had on each. I'll try and stick this back up in the corner here. So we first have 2 points of reference in the wheat bran and Purina chicken starter, the amount of percent crude protein available in the feed. Then there is literature data of what the mealworm percent crude protein is from other literature sources. And then that is compared with our 3 generations of the P0, F1, and F2. Within each generation, the percent crude protein decreased. And I'll talk about the conclusions here in a slide or two. Next, talking about the average mealworm percent crude fat and the effect that generation had. Again, we have the wheat bran and the Purina chicken starter as the feed values of available percent crude fat there, with literature data from the same sources as well on the mealworms to signify what their percent crude fat was. And then we have our 3 generations of the P0, F1, and F2, where again, there was statistical significance in generation, not in treatment. And with each generation, the percent crude fat increased. And lastly, talking about the average mealworm percent dry matter and the effect that generation had on each. So again, there are 2 points of literature data here on mealworms and what their percent dry matter was. And this was compared with the 3 generations as well, where there was again no significant effect of treatment, but there was a significant effect of generation. And so with each generation, the average percent of available dry matter increased. So what does this all mean when we put these things together? So there was no effect of treatment on percent crude protein, percent crude fat, or percent dry matter, meaning the differences in the abundance, blend, and sufficient treatments had no statistically significant effect, meaning there was no differences between the control or the styrofoam fed treatments, which leads to the conclusion that styrofoam does not affect nutrient composition when applied for treatments. However, there was that generational effect. And generation does have a statistically significant effect on percent crude protein, percent fat, and percent dry matter. And this effect is likely due to the continued use of Purina chicken starter, which is a nutritionally complete and balanced diet for an omnivore, which mealworms are an omnivore. These values differ from literature data due to feed differences of chicken feed versus the industry standard of wheat bran. To dive into system dynamics modeling, we'll first start with the population model. This population model was co-built with Ivan Taylor, and the goal of the model was to create a lab-driven model that could display behavior over time in each of the population in each treatment, and using lab data collected from 3 generations in order to determine what the optimal ratio of chicken feed to styrofoam for population growth over the long term. The population was estimated over 1,000 days, or a total of 10 generations. So here are the results of the population model. And I want to point your attention to the x-axis here, which is days, and then the y-axis, which is number of larvae. Each treatment was parameterized from lab data and used to create and simulate this 1,000-day population. Over that 1,000-day period for the models, there was no clear expanding or dampening of any oscillating behavior. It was what's considered sustained oscillation, which helps explain how many mealworm farms, both small and large, are often managed. This is due to mealworm farmers typically allowing 5 to 10% of their larvae to pupate each breeding cycle. So I'm going to point out abundance 32 here, which is this brown line as we go through, which has 5 oscillations or peaks and valleys. And then we have blend 48 and sufficient 32, which has 3 large peaks and valleys. So this population behavior can detail how many larvae are being harvested over a 1,000-day period. Oops. Back here. So this is the harvest of each treatment simulated over that 1,000-day period. And there is continuous harvesting of larvae throughout this system, in which one would probably assume the abundance treatments would seem to have the highest harvest amount because of their relatively stable harvesting periods. However, sufficient 32 is the largest number of larvae harvested, which is this pink line up here, and then blend 48 being the second largest, which is that red line, and abundance 32 being the third highest in terms of amount of larvae harvested. Sufficient 32 and blend 48 being greater than abundance 32 is due to that severe increase and decrease in larval population, referring back to those 3 big peaks and valleys. Now, while not all parameters are considered within the population model, the supply chain model adds another data point in determining which treatment ratio is satisfying all of the objectives. So after optimal feed and rearing conditions were determined, a farm scale model of a larger system was determined to and designed to explore feasibility, profitability, and sustainability of a large scale mealworm farm. The model utilizes a sustainable closed loop conceptualization such that waste products are utilized and made into products. The supply chain model does utilize many assumptions based on industry standards, personal experience, and reasonable estimates. System dynamics is uniquely suited for this type of hypothesis testing and establishing and testing parameters that are critical for the system, but for which there may not yet be known established values. This is an image of a mealworm farm. This is about a 120-pound or so mealworm farm of what it would look like. It doesn't take up that much space. This is very, very small, so mealworms are very space efficient. And then brings us into our next causal loop diagram of what a traditional mealworm farm would look like. And I'm going to walk us through each step of this. So we have incoming nutrient-rich food, which goes into the mealworm farm. Mealworms we then know produce frass. Frass can be used as a fertilizer for agriculture. We can then take agricultural food waste, which many insect farms and mealworm farms are starting to incorporate more, back into as a nutrient-rich food to the mealworm farm. We also have mealworms as an output from our mealworm farm. They help generate some revenue. We also have associated costs with running a farm in the form of rent, heating, electricity, water, and labor. And all of those boil into our total profit calculation. This is another causal loop diagram, but it is expanding upon a traditional mealworm farm and adding in the styrofoam processing to the mealworm farm. So the mealworm farm gets an additional input of incoming styrofoam waste. And there's an associated collection fee with that for the farm taking in the styrofoam waste. They will be getting paid a certain amount of money for taking in that styrofoam waste. This also brings up the potential excess styrofoam flakes or other excess contamination of styrofoam floating out of the farm system that can be contained and can then be compressed into bricks, which can then be sold to sorry, I don't have it there, which can then be sold for revenue. So I now want to look at the supply chain model. The supply chain model is, again, this is a causal loop diagram of the supply chain model. And again, the incoming styrofoam waste and that collection fee is the money that we are making with this. But this breaks the outputs of the mealworm farm down further into defatted mealworm powder or more concentrated protein, mealworm oil or more concentrated fat, and again, frass, which would be going into agriculture. But it's simplified here for clarity of this. And that's that defatted mealworm powder and mealworm oil. From this model, we can run several simulation scenarios, including simulated styrofoam consumption for each treatment. So this is the simulated styrofoam consumption by the mealworms. These numbers were first derived in a per mealworm per day on a gram basis, and then scaled up to a pounds per month basis based off of how many worms are in the system. So the supply chain model assumes a 200-pound larval operation over 60 months of operation. There are no mealworm industry standards, so a conservative 200-pound larval operation was chosen based off of some commercially available information. And again, these rates scale on a linear basis within the model. So there is a point of diminishing returns at extreme large scale operations that are outside the bounds of this model. So the rate of styrofoam consumption by mealworm in the treatment that had the highest consumption rate was the Abundance 32 at a rate of 11.3 pounds per month. And it was used in the 4 following scenarios. The first scenario here is the competitive pricing scenario, which is this bottom blue line here. And so each of these other scenarios were chosen at a realistic look at how the farm could be operated and to show the vast differences in profits based off of which parameters were changed. So it's currently assumed that a 200-pound mealworm facility can only take in around 25 pounds of styrofoam a month. And in the competitive pricing scenario, which again is that blue line down here, defatted mealworm powder is sold for $4. Mealworm oil is also sold for $4. Frass is sold for 50 cents a pound. These are all on a per pound basis. And styrofoam collection is charged at $5 a pound. Again, with the assumption that all of these products are sellable and a mealworm, with that, the mealworm facility of this size, of that 200-pound, with those competitive pricing to compete with whey and soy and pea proteins on the market, is a loss of around $400 per month. Considering that's an undesired outcome, I wanted to look at another scenario, which is the scaled scenario. In this scenario, only one parameter is being changed, and that is the size of the mealworm farm. Changing it instead of a 200-pound larval farm to an 800-pound larval farm. So this is a quadrupling of the farm output in terms of its products. All of the other pricing of the defatted mealworm powder, mealworm oil, frass, and styrofoam collection remained constant with the competitive pricing. Again, the profit falls within the range of $93 to $138, depending on how much is harvested per month. And while these are profitable, a profit of $100 a month is not interesting for a business. So we're going to look at the next scenario here, which is called the premium pricing scenario. And in this scenario, it is changing the price points of defatted mealworm powder to $12 a pound, as well as charging $25 a pound for styrofoam collection. So really charging a big premium for these services that are being provided. And this brings the profit up to, of the 200-pound mealworm farm, to a range of $308 to $320 per month, depending on, again, harvested amount. And currently, the market price for defatted mealworm powder and cricket powder and other insect powders is around that $12 mark, which isn't competitive to whey or pea or soy protein, but that is what it's being currently sold for on the market. So this is a potentially realistic scenario that could be implemented to raise profits of the mealworm farm. And lastly, our fourth scenario here is a styrofoam innovation scenario where all assumptions of the farm size and the amount that everything is being sold for, of the $4 for mealworm oil, $4 for defatted mealworm powder, 50 cents for frass, and that $5 collection fee for styrofoam. The change in variable here is instead of only taking in 25 pounds of 10xing it and taking in 250 pounds of styrofoam a month. This raises the profits up to around $718 to $729, again, depending on how much is harvested per month. And whether a mealworm styrofoam consumption could actually handle this amount of styrofoam is a currently unanswerable question. However, it may be possible under better rearing conditions, as I mentioned before, of better humidity and consistent temperature, or an increase in styrofoam consumption through other novel techniques. So for our conclusions, first, we found that there was no styrofoam in frass of any treatment or any generation. We did find a generational effect of percent crude protein and percent crude fat, as well as percent dry matter. And it was determined that the Abundance 32 was the best overall treatment. That was because it had the largest available percent dry matter and subsequent harvestable weight, and it wanted the largest harvestable amounts over that 60-month simulation. It has one of the highest simulated populations and mean populations. It did produce the largest styrofoam breakdown, and it was within reason for other treatments for profitability. So the main conclusion derived from all aspects of this research were that if you give mealworms more styrofoam, they will consume more styrofoam than also being fed Purina chicken starter. Now, industry standards on size and capabilities of farms do need to be determined, and whether insects can produce well at scale. It is yet to be shown that insects are a long-term valuable or successful product on the wholesale or retail markets. And it has been yet to be shown if they can sufficiently compete with whey, pea, and soy proteins. The nascent market and lack of available information of current business profitability proves any analysis of this challenging. Some of the next steps that can be taken is around addressing the mealworm consumption concern in the view of microplastics, as well as potential FDA regulation. So one of the main concerns is microplastics being present in the mealworm bodies, and there are not a sufficient amount of studies to back up that mealworms would be safe to consume based off of microplastic presence. So mass spectrometry or FTIR image analysis or any other type of particle count or microplastic analysis on mealworm bodies would be very beneficial to further addressing these concerns for consumption. Interestingly as well, the FDA also has a set amount of styrene, which is the chemical component of styrofoam, that is allowable in foods. It wasn't until 2018 that they banned, yes, it wasn't until 2018 that they banned styrene as a food additive. That is 4 years ago that we banned styrene as a food additive. On top of that, there are still minimum amounts of styrene that are allowable in food. And it's been detected that there are natural styrene occurring in beef samples, coffee beans, strawberries, and cinnamons, as well as associated other vegetables, fruits, and beverages. So mealworms have yet to be thoroughly tested whether or not they have styrene present in their bodies, as well as microplastics, but previous FTI results from my undergraduate study did show that they would be likely to fall under the FDA styrene concentrations. The next modeling step that can be taken is to first connect the behavior of the population model to influence the supply chain model. This would help researchers and potential investors better evaluate the sustainability of a styrofoam decomposition mealworm farm. As well as improving upon both models can also be done through running lab experiments longer than 3 generations with optimal temperature and humidity to see if the simulations of those 3 generations over that 1,000-day period hold true in the long term. And this would allow for further statistical validation of trends that were being displayed. This can also be done through another modeling step of incorporating a circular economy mealworm farm to the equation. So again, this is another causal loop diagram where we have our same inputs of styrofoam waste and nutrient-rich food into the mealworm farm. But what we are adding is mealworms going to aquaculture. Whether this is whole mealworms being sold or the broken down products of the defatted mealworm powder or the mealworm oil going to aquaculture, it allows a place for them to go to feed fish in these aquaculture systems. And aquaculture generally uses large amounts of styrofoam shipping material, which can then go back into the mealworm farm system. Again, we have frass going into agriculture and taking agricultural food waste into our mealworm farm. And any potential excess styrofoam flaking can go into brick compression to be put back into the plastic life cycle. This would close the loop instead of there not being a closed loop system. We are traditionally not very good at closing loops of systems, and there is a real potential for a mealworm farm to act in a closed loop system. So our closing thoughts here. The research tackles only a small portion of the plastic problem, only styrofoam. And insects can help in not only breaking down styrofoam, but also provide additional nutrition to undernourished populations that can help, as well as push the scientific community towards finding more solutions for plastic management and sustaining increasing populations. I want to thank everybody for coming, and we will now transition into our Q&A section. Yeah.
I have a few questions.
Okay.
I'll repeat the question after she says.
Okay, we'll do.
So, partly on sort of the methodology on how you were collecting, measuring protein, and were you grinding up the worms and then measuring it as a whole worm, or was it?
Yeah, so the larvae are ground up, and it is through, I'm just checking my notes here so I get the name right. It is through Caltech analysis on percent crude protein and sock slit ether extract for percent crude fat, which basically you do a drawing of the worms to get rid of all their moisture. And then we sent it off to a lab called Zuquarius, where they ran these tests of ground up worms through these different methods.
So what is the dry, what was that measure? I guess I'm not sure I understand what that is.
Yeah, so it's a
dry matter.
the dry matter.
Be sure to repeat the question if they're asking.
Yes, sorry. The question was, how does the process of percent crude protein, percent crude fat, and percent dry matter happen? So for the percent crude protein and percent crude fat, right, they grind it up after it's been dry mattered. The process of dry mattering is removing all available moisture, so you're getting just its true dry weight instead of having any water weight in there.
And that's ground up and then you measure protein. Okay.
Yes, and then that is ground up, and protein and crude fat are measured after that.
And I'm also curious about your modeling. So you showed causal, like you said, causal diagrams. The diagrams, are those just conceptual maps? Did you actually model in like Stella, or did you model in Excel? I remember you saying by Excel. So.
Yeah. So the question was, were the models just done in a causal loop diagram, or were they done through modeling software? Right. That's.
Well, I know you use modeling software, but did you use Stella or did you use Excel? Like, what did you?
Yeah.
use?
So the question is, which modeling software did I use? Excel was used for the population model, and that was co-built with Ivan Taylor to do calibration through taking lab data to create a calibrated population model. That was then transferred into Stella to provide the graphics that we're seeing here. The supply chain model was solely modeled through Stella Architect, and it's a very large, messy graph, so that's why I did the causal loop diagrams instead.
All right, so we've got some questions here from Jen. You described the humidity as lower than ideal. I may have missed it, but what is the ideal humidity, and how might that affect the life cycle?
Yeah, that's a great question, Jen. Thanks for chiming in. So the potentially best and the literature best optimal humidity is around 80% relative humidity. There is some nuance there depending on which stage of life they're in and different things like that, but 80% is the generally accepted literature optimal humidity. And so how the lower humidity could have affected the mealworm life cycle and feeding is a great question, because that is one of the confounding factors, that as much as humidity and temperature and the location of all of the bins throughout the 300-day process was tried to be controlled as much as possible, those were just not feasible things. So when looking at other research and other industry standards of life cycle, they can be shorter in some cases. There also is the addition of carrots or potatoes as a moisture source. And that was not done in this study to get a flatline nutrition analysis of just the effect of chicken, the Purina chicken starter and Styrofoam. So it definitely can affect it. I won't say whether or not it would have increased or decreased because the literature has a wide range of where values fall. All of the total time of life cycle for this study fell within the average of all research studies at around that 140-day period. It can go as fast as 60 days and it can go as long as 260 days. So there's a very large time amount that the life cycle can be depending on their conditions. Move on to the next. I can highlight, Jen, if you have any further. I'm going to participants.
Jen's got about 5 questions.
Yeah. All right, Jen, I'm going to give you ability to talk through so you can, since there's questions here, in case I don't clarify anything well enough. Oh, it would help if we had volume.
Oh, no, I wasn't speaking.
Oh, okay, cool. We can hear you.
Yeah, thanks, Brenden. Go ahead and, I mean, I didn't know if you wanted me to repeat the question or if you only want me to speak if I have a question about what you're going to say.
I'll hit the questions that you have here in order, and if you want to stop me at any point, just chime in, and I can try and clarify further.
Okay, and if people have questions in the audience, don't feel like you have to do mine all at once now.
Okay. So for your question, what would the FTIR scan have looked like if there was Styrofoam present in the terms of small versus large residual contamination? The spectra would be around and I can actually go to, let me try and go back a couple slides here. I can point out where on here it would be. This is backwards. Got to go through all the animations here. Just as a better visual representation. I should have had an additional slide for this. All right. Wow, really going back. Okay. So let me move these things out of the way here. So for FTIR of Styrofoam or polystyrene, it would have very sharp peaks at the 3200 wave number, 3100 wave number, as well as the 2900 wave number with that small peak. So there is a matching peak here, but that is just a carbon-carbon bond, which that carbon double bond is present in both polystyrene or Styrofoam, as well as in chicken feed and frass, which is made up of purines and uric acid, so they share functional groups there. So this is why I used industry software to further try and get an analysis of matching up to a pure substance. And the absorbance levels are also much lower for the polystyrene, or for the Styrofoam as well. Instead of going up to this 0.08 up here, they are in this 0.02 to 0.04 range across the board for matching up spectra.
So you feel that if it had been there, you would have been able to detect it?
Yeah, I did a baseline FTIR analysis on 2 different samples of polystyrene, as well as using what the industry database has to compare against it to see if there were any matches. And the software, as well as any matching up of peaks, was only in this 2900, 2800, and none of the other peaks were matching across the board. So the likelihood of them being similar substances or similar peaks is relatively low from FTIR analysis. That doesn't mean that further analysis, though, could be done to detect nanoparticles or micro particles of plastic. I'm not 100% positive on all of the limitations that FTIR has when analyzing. If there is, 1 part per billion in there, I don't know if it would pick it up in terms of that. But that is a good question, and further frass analysis could be done to detect if there might be any of those nano or microparticles within the frass.
The next question there had to do with the mealworms themselves. You mentioned that you had not been able to look, or didn't have the time to look maybe at the actual bodies. And I was just thinking, like, if you'd seen that there was some, if you knew what sort of a clearance time might be required, and if that would make sense for your farm proposal to have a clearance period.
Yeah, so if the nano microplastics could be cleared through that, there's unfortunately not a whole bunch of data on just the presence of them, if they're even bioassimilated or anything to that manner.
Yeah.
However, most of the plastic research studies have shown that mealworms, when they consume it, it's around a 24 to 48 hour period when they then ingest the material. So through that, there is a potential where if you fast the mealworms before turning them into feed consumption to make sure all of that frass is out, that there is potential that they could be clean in terms of any styrofoam, nano, or micro particles. And in my undergraduate work, I did FTIR the mealworm bodies, and there was no signature of styrene or polystyrene, but that is one study out of countless that could be done, and other methods of, I don't even know if FTIR on the mealworm bodies is the right analysis to do for looking for nano or microplastics. Something like mass spec or other imaging might be a more efficient way of coming to a conclusion on that.
Cool, thank you.
Awesome. All right, let's see. Next one here.
Do the last one. And then come back to that fourth one.
Okay. An intergenerational difference in protein and fat with averages with no error bars. Yeah, so the statistical significance between them for the percent crude protein and percent crude fat. Sorry, mouse is. Were the parental generation was statistically different, or sorry, the generational difference. So the parental, which was the P0, was statistically different than both the F1 and the F2 for percent crude protein, and the F1 and the F2 were not different from each other. And that is consistent for both the percent crude protein and percent crude fat, as well as the percent dry matter. A trend of the parental being statistically different of a generation to the F1 and F2, which are statistically similar to each other.
Thank you. All right, that fourth question, then I won't bother you with more.
Cool, okay. Indicated in the SOE 531, concern about generating microplastic pollution by feeding styrofoam chunks to mealworms. How are you feeling about that? No. Do you think the bricking will sufficiently capture those particles? Yeah, it's a really complicated question and problem of the frass sifting mechanism with the 1/50th inch mesh really did seem to eliminate any visible to the naked eye particles from being within the frass sample. However, I am still worried about potential micro or nanoplastics being in there. I did take representative samples to the best of my collection ability, but I didn't analyze every single, 100 or 200 grams worth of frass that was present. So I am feeling better about it, but there are a bunch of other potential concerns of when you're actually doing this at scale and you have to have an HVAC system. Is the airflow going to blow styrofoam particles around? Is the movement of bins going to blow styrofoam particles around? Will the transfer of cleaning them cause these problems? So the bricking will sufficiently solve the, once they are captured, problem of turning them into something that can then be used. The capture mechanism is something that I envision as a very clean room, and having, similar to a lot of other places that do particle captures in places, and implementing something like that to capture particles so they can then be processed into something. So you have to have a lot of different securities in place in order to ensure, because styrofoam is so light, and we know that plastics, especially styrofoam, can be airbound. It becomes a real concern if they do become airbound on how to efficiently control for them. So it is still a concern. I think some of the concerns have been addressed, but there are still many more that I don't have a solid answer or solution to, that I wish and I did, but I think they just need to be further evaluated to see if they if those mechanisms actually work.
Thank you.
Yeah, thanks for all your questions, Jen. I appreciate it. Are there any other last minute questions here?
Nothing in the chat.
Yep, I think we're good there. Any last questions from audience members? Yes.
What is your favorite mealworm recipe?
Ooh, I like that. My favorite mealworm recipe. Okay, well, there's two. I know one that, like, it's a little bit hard to get over the fact of it, but it's really good. There's this woman in the Oakland, San Francisco Bay Area, and she fries up using some traditional oxalon techniques. I think that's the region of Mexico that they derive from, where she sells whole mealworms, but they're fried up and as a whole mealworm, and with like corn nuts and other stuff like that. And you just kind of eat them like you would, like, I don't know, ball game stadium food. It's a little bit weird to get over the I'm eating a full insect, but they do taste really good. My other favorite would be if you grind them up and putting them into like a banana bread or any type, like a bread like that. It's actually really good because the flavor doesn't come across too strong. It's very like nutty and cinnamony flavor, so it can help augment the, like a banana bread or I don't know, some type of bread that is good with those flavors. So yeah, I think those are my two favorite.
I'll give you one. So you were comparing, so in one of your treatments, you had 6 grams of styrofoam to 192 grams of chicken starter.
Yep.
By volume, how does that look different?
Oh, the volume of 192 to 6
Yeah.
grams. Yeah, I think I can, let me find the image. Let me go back. It's this image here. I know there's no chicken feed in here. Actually, wait, I have one on my supplemental slides. Hold on, let's do it this way. Let's just scroll. So this is a, this closing thoughts, hold on, this one right here. This is partially broken down mealworm, or partially broken down styrofoam, and chicken feed is buried under all of these exoskeletons right now, but this is a kind of visualization of what it would look like of the bin, and I can draw with my hands of a bin is about this big.
I just mean, if this much styrofoam,
Oh yeah.
is it this much chicken feed for weight to volume?
Oh, I see. Yeah, so the chicken feed amount would probably be 192 grams is about like 4 full handfuls of chicken feed versus 6 grams of styrofoam. You can fit a block into one hand. So that's like a difference of it. I should have had a picture of a clean
It's just a funny
box.
thing to correlate.
Yeah. So I agree. Yes,
I have a question. The worms that we used on your honors project,
yes.